So, welcome. I'm going to walk you through how to essentially create a protein. So, I don't often recommend that you just go through the motions first, but in this case, because transcription and translation are processes, sometimes just actually getting in and doing the process can help, and then you can layer in the vocab later once you get a sense for what's going on in terms of the big picture. And you can deal with the nitpicky, like all the players and things like that afterwards. So, what we're going to start with is we're going to start with our DNA. Now, keep in mind, DNA is a double-stranded molecule. It's a double helix. But what we're doing is we're only looking at one strand of that, the template strand. So picture if you have your double helix, it'd be like if you unwound it and you had a ladder, you took that ladder and you sawed it in half and you just looked at one half of it. So that's what we're doing right now. We're just looking at one half, the template strand. So when we have DNA written out this way, that's why it looks that way. Also, it came up in class, like what's with the letters? Well, if we look at what this is actually saying, it's saying that if we have this strand of DNA, the first nucleotide we're looking at has a nitrogenous base that has a thymine, that is a thymine. The next nucleotide that we're looking at there has a nitrogenous base of adenine. And the third nucleotide that we would be looking at down in the sequence would have a nitrogenous base of cytosine. All of that is a mouthful, so scientists shortened it. They can represent that entire nucleotide just based off of the first letter of the type of nitrogenous base that makes up that nucleotide. So that's what we're looking at here. So if we look at that, we start with our DNA. We know that our DNA is too big and too important to leave the nucleus. So what we need to do is we need to transcribe it. In other words, we need to make messenger RNA, because messenger RNA is going to be just the information we need just for the one particular gene that we're looking at. It has the instructions for the single protein that we're trying to build. And that's going to be small enough to be able to slip through the pores of the nucleus and actually travel into the cytoplasm, where it'll meet up with a ribosome, which is like the protein building factory of the cell, so that that protein can actually get made. So that's what we're doing. So we have to remind ourselves of these rules. If you have a DNA thymine, it's going to pair with an RNA nucleotide that has an adenine as a nitrogenous base. Let's simplify that for these other ones. DNA A pairs with an RNA U, DNA G pairs with C, and C will pair with a G. And practice makes perfect. The more you do this, the more that will just become innate, and you won't have to worry about quote unquote memorizing it, because it will just be memorized because you've done it so much. So if we look at this next one, we have a little text box for ourselves. A DNA cytosine is going to pair with an RNA guanine, and then an adenine, and then a cytosine. And if we keep going, we've got an adenine will pair with a uracil, because anytime you're tempted to put a T, like if you were doing DNA to DNA, you're going to put a U. For chemistry reasons that we're not getting into, RNA just doesn't have thymine as an option for nitrogenous base. Instead, it has uracil. So uracil, though, is able to bind to a DNA adenine. So that's why we get A pairing with U. C, I'm going to pair with G, U, and you just kind of just get into a rhythm with it. You become pretty good at it. I don't recommend speeding through it, though, or you will make silly mistakes. And always just check your work. Dun, dun, something like that. Then you've just made an mRNA strand. So the next step is to actually build a protein with this. This is, in theory, the instructions, the shorthand for how to build a protein. So that's what you got to do. We know that proteins are chains of amino acids that then get later packaged and processed into actually becoming what we know of as finished proteins. So in order to, the first step of that, to make that polypeptide chain, we need to know the sequence of amino acids that get put together. What we do for that is we use a codon wheel. So now picture you're in the ribosome. Let me erase all of this here for us. Start fresh. Boop, 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 boop. Okay, so picture you're in the ribosome. The ribosome is this protein building machine, and it's reading the mRNA one codon at a time. The same way you would read a sentence one word at a time, it's reading it one codon at a time. What's a codon? A codon is 3 nucleotides, 3 mRNA nucleotides. So in this case, here's our next codon. GAC is our codon. So what we would come to is our codon wheel. Start in the middle and work our way out. It was, GAC. So what I'm going to do is I'm going to get a highlighter to help us out here. Start in the middle. In theory, just thinking about it. Cool. GAC. So it's telling us that our first amino acid is going to be aspartic acid. And that's a lot to write out, especially in such a limited space. So I'm going to abbreviate it ASP. Good enough, aspartic acid. There's other ones, so there is like an official way to abbreviate, but this is sufficient for us. So that's our first one. And then if we look, it gives us instructions for how to kind of color it. That's just our creative way of illustrating how to build this protein. So in this case, it's telling us red and down. Red and, let's see, down. It's confusing because it's green, but that's okay. Next up, we've got UGU is our next codon. So if we come over here, I'll use a different color for us. U and GU, it's telling us our next amino acid is cysteine, and then we're going to color green and to the right. So we'd come over here, and in our chart, we'd keep track of that. Green and to the right. Ooh, it's going to go in this part of it. I need to write what the amino acid is. That's the important part. It's cysteine. And I'm just going to abbreviate it with the first 3 letters to make our lives a little bit easier. Let's do one more. GUU. So G, starting in the middle, U, valine is going to be our next one, and it's telling us orange and up. So valine, VAL, and then orange and up. I don't know how to draw that up, so we're just going to have to draw it. Doop, and up. Yours will look nicer than that. So now comes actually drawing it. And again, this is our creative way of trying to illustrate how to draw the protein. In reality, we know that it's not exactly like this. Like there's charges involved, there's polarity involved, and they're not like colors nicely stacked on top of each other in boxes. That's okay. So it starts with, our first one was given to us. It was black X. So that's kind of like a reverse pirates map here. We've got X marks the spot. Cool. Then it tells us red down. So I'm going to take red. I think highlighter might show up a little bit better. Let's try this. And freehand, there we go. I'm going to color this box red. And then it says the next one is green, but over to the right. So I'm just going to pick up from right where we left off and color this next box green. And then it says orange and up. So I'm going to find orange I don't have. Let's see if there's an orange. Have an orange here? More colors? All right. Good enough. There we go. What? Orange. Save. There we go. I just want to show you one more. There we go. Okay, orange up. Cool. So you would keep going through that pattern, and eventually you should get a particular shape. And that's it. That's you being a ribosome, building this protein based off of the codons, one codon at a time, adding one amino acid at a time, which is you coloring. Good luck.